How are you?
Yeah, I'm absolutely fine. How about you?
Very fine, very fine.
Yeah, thank you so much. Actually, I could just reach the office of our meeting for
Oh,
IPP.
thank you. Sorry to disturb you. We'll make you. Okay,
Right. And I'm ready from my side. So you can just tell me whenever.
okay, no problem. You just be ready, huh?
Yeah, yeah. Thank you.
Okay. Um, Vandana, madam.
குட் ஈவினிங் திருமலை சார்.
குட் ஈவினிங், மேடம். எப்படி இருக்கீங்க?
நல்லா இருக்கேன் சார். நீங்க எப்படி இருக்கீங்க?
நல்லா இருக்கேன், மேடம்.
கங்க்ராச்சுலேஷன்ஸ்.
இல்ல, அதான் ஒரு கிளாஸ்ல இருந்தேன், அதனால எடுக்கல. சாரி.
ஓகே ஓகே.
ரொம்ப சாரி.
ஒன்றும் பிரச்சனை இல்லை. நான் தான் லிங்க் எதுவும் வரலையேன்னு கால் பண்ணேன்.
Sir. Okay.
லிங்க் இன்னைக்கு தானே அனுப்பிச்சிடணும்.
Hello, sir.
சரிங்க, மேம். வந்தனா மேடம்.
Hello.
வந்தனா மேடம். வந்தனா மேடம். மேடம். வந்தனா மேடம். வந்தனா மேடம். சாரி, வீட்ல இப்பதான் போய் ரீச் ஆனீங்களா? இப்பதான் ரீச் ஆனீங்களா?
கேக்குதுங்களா?
கேக்குது. என்ன கேக்குது? என்ன கேக்குது?
இல்ல சார், இப்போ 15 minutes ஆகுது.
அதான் வந்தனா மேடம். 55 ஆச்சு. புரியல.
15 minutes ஆகுது.
மேடம். ஓகே. வந்தனா மேடம்.
Okay, sir.
Veluram sir is also with us now.
Sir, shall we start, sir?
Okay. How many participants right now? எத்தனை participants பாருங்க.
Right now 17 is there, sir.
Now let them
17 வந்திருக்காங்க sir.
இன்னொரு 4 நிமிஷம் இருக்குல்ல. இப்பதான் ஜாயின் ஆகுறாங்க. ஜாயின் ஆகுது.
Yes, sir.
Veluram sir, good evening.
Good evening, sir. Good evening, sir.
our speaker வந்துட்டாரு, ஆல்ரெடி வந்துட்டு வெயிட்டிங்ல இருக்காரு.
Okay.
வேல்முருகன் சார், குட் ஈவினிங் சார்.
Good evening, madam.
மலர்கொடி.
Hello, madam.
ஓகே சார், கங்க்ராட்ஸ் சார்.
Thank you.
ஸ்டார்டிங் ஆஃப் தி FDP.
Thank you, madam. Credit goes to Dr. Thirumalai Kumaran.
ஓகே சார்.
இருபது பேர் தான் இருக்காங்களோ?
சார்
யூடியூப் லிங்க் போயிடுச்சாமா? வந்தரா மேடம். யூடியூப்.
எஸ் சார், கிருத்திகேஷ் கொடுக்காங்க.
ரைட், ஓகே. ஒரு 50வது வரட்டும், வந்தப்பறம் நம்ம ஸ்டார்ட் பண்ணிக்கலாம்.
ஓகே.
வந்துரும். Vinodini madam, good evening. Prakash ji, Prakash,
Yeah.
let us
Yeah.
start.
Yeah, we can.
It is 6. Now it is 6. Shall we start? Sir, Velmuru sir, start.
Yeah, please, sir.
Right, okay. Start, please. Madam, start.
Okay. A very pleasant evening to everyone, and thank you for joining us. I am Mandana, lecturer from Savita College of Pharmacy, CIMATS. I will start this session with a beautiful quote. All that man needs for healing, health and healing has been provided by God in nature. The challenge of the science is to find it. First and foremost thing, I would like to thank our Chancellor, Vice Chancellor, Director, and the Registrar for providing this opportunity to conduct the 5 days online FDP. I also thank our principal and other faculty members for their cooperation with us. Now I would like to call our beloved principal, Dr. Velmurugan sir, to welcome the gathering.
Thank you, Vandana. Good evening, one and all. Thank you to each and every one of you for being here with us today. We are very much pleased to be able to welcome those of you that have been with us for a long time, as well those who are new to the group. I welcome the resource person of the day, Dr. Prakash Itankar. Today marks our first day's National Online Faculty Development Program on modern approaches in herbal drug development, and we are proud to be able to host this event today here at this online platform with all of you. I hope the session to be productive enough, and please do enjoy the session. Thank you. Over to Vandana.
Sir, to welcome once again for this online FDP, which is going to be held from 26th today onwards up to 1st of December. In this event, around 750 participants are joined from various states in India. So I'm very happy to introduce my friend and my society friend, Itanga, Prakash Rambanji Itanga. He is the professor of pharmacognosy and director Technology Park at Nagpur University, Nagpur. He is known to me for the past 10 years because of Society for Ethnopharmacology. We worked together for the past 10 years, and he is a very good entrepreneur also for making different events, and he is helping for industries to solve the problem also. He's having experience of about 20 years in teaching, including 6 years of industrial experience, and he handled around 43 research projects, which may be solving the problem in the herbal drug industry also. And he guided around 105 M.Pharm graduates, and PhD he guided around 9, and students pursuing PhD is around 8 under him. And he has about 73 research publication in national and international journals, and he has given innovative lectures around 125. That includes Society for Ethnopharmacology also. Is one of the key speaker in the Society for Ethnopharmacology, and he was awarded with the various awards like Dronacharya Award in the year 2022. Dronacharya Award he has received in the year 2022, and I can proudly to say his university, he is the chairperson for conducting IPC 2023. He is hosting the IPC, and also he has got a Best Teacher Award, as well as Ethnopharmacology Outstanding Service Award in the year 2018 and 2015. He has got these 2 awards from Society for Ethnopharmacology, which is there in India for the past more than 25 years around. And he has done different research projects under natural products. His area of interest is in Panchakavya, pairing Panchakavya. That is his area of interest. And over to you, Prakash. Very welcome, warm welcome to you.
Thank you so much, Dr.
Before that, we wanted to felicitate Vandana Madam, please. Vandana Madam. Vandana Madam.
Yes, sir.
Please.
Yes, sir, I can hear you.
Please, over to you, Madam. Madam, over to you.
Yes, sir. On behalf of the Savita College of Pharmacy, we wish to felicitate the speaker with the sample of medicine. Yes, sir. On behalf of Savita College of Pharmacy, we wish to felicitate the speaker with the sample of medicinal plant, which was planted in our medicinal garden. I was sharing that picture.
Yeah, thank you. Thank you so much.
Thank you, sir.
Thank you, Prakash.
Thank you, ma'am. Thank you. I would just present my thing. Just.
Okay, sir. Over to you, sir.
Just a minute. I'm just putting my PowerPoint presentation here. Share. Is it visible?
Yeah, now it is visible. Yes, Prakash. Please.
Yeah, I'm putting it on full screen now. Is it visible?
Yeah, visible. It is visible.
And is it moving now?
No, it's not moving.
It's not moving. So it means I have to use, because many a times it happens. And in that case, I will just increase the screen size.
Yeah, no problem. That's also you can do it.
And so very good evening to all of you. And I'm extremely thankful to management of Savita College of Pharmacy and my dear friend Dr. Tirumala Kumaran sir for giving me this opportunity to address all of you in this, those who have joined 5 days National FDP on modern approaches in herbal drug development. So I took up, a very light kind of a topic here, and the kind of thing that I'm going to share with you is something which all of you would connect with, and you would just go into the kind of experiences I went through, and through which I gained a lot of knowledge, and I could become, something who delivers to the needy in this particular domain. We do help, so many industries, the micro, small, medium entrepreneurs, or budding entrepreneurs, or even the bigger industries. We are helping them in resolving their practical issues. So this all has become because of the approaches that we have taken in the herbal drug development and its establishment in our day-to-day life. So I belong to a branch that is pharmacognosy. All of us, we know that this is the field which talks about the drugs coming from natural origin. However, the inspiration for me to work so hard and particularly in this herbal drug is because of my industrial experience, which I owe after immediately passing out my master of pharmacy. I started my job in industry, and industry has taught me many other things which many of us in academic institutions do not touch. And one amongst that is using the time appropriately for many developmental activities, including the societal work. So I would just like to first take you to my next slide, and this talks about something which all of us believe in, and that is called as the cosmic life force. Usually, whenever we think about us, we believe that we are quite a learned person. We have grown up, and now we are in a position to tell others and teach others and preach others. And then, along with us, starts getting attached the factor that is called as ego factor. So I just wanted to bring all of us on a baseline that looking into the whole Brahmanda, if you see, the space for our own earth is just like a dot here in one single Brahmanda. So imagine when our Rishi Munis were talking about the Ananta Koti Brahmanda, then you can imagine where our place lies. So our place of earth itself is minuscule in this whole infrastructure. And then what everything is governed by, and we usually believe that it is something like we call it them as almighty. So the cosmic life force, or the kind of a energy which governs us all, including complete earth, needs to be understood first, because we are talking about the drugs coming from natural origin. So what are those laws of nature because of which all this Brahmanda, these graha nakshatras are being taken care of? So I would just like to address you with a very wonderful definition I came across while studying many books, that this particular force is called as a life force, which holds everything in the cosmos together, and it can be defined as a natural, intrinsically automatic, self-regulatory, systematized order endowed with the universal laws of nature. So it means that एक ग्रह दूसरे ग्रह के साथ में they are not colliding. Everything is properly hanged up, and everywhere there is a kind of a function. Now that function on Earth, if we zoom in, can be read into the kind of biodiversity that is being bestowed upon us. Now this cosmic law of nature on Earth can be looked into in the various forms, like we as the creatures, the animals, we can say, the plant world, and then the animal world. So if you look into more focused into this, we can see the picture like this, that on the surface of the earth we have the vegetative strata, we have got the animals, and we as a human beings, we are also there. Now if you peep into the past, we can really understand that how the humans have really made use of the flora on fauna and the kind of facilities we in the form of, say,
looked into
the clothing
in the various forms,
the
like
resources
we
given by
as
the
a
nature, the shelter that he
creatures,
prepared, and the kind
the
of
animals,
food,
we can say,
which
plant
he could
world,
choose
and
from the
then the animal
available
world.
biodiversity. The smartness available in the human
So
being,
if you look
has
into
really made wonders. And then
more
we
focused
started understanding this particular capacity in the form of our brain.
into this,
So
we
it
can
is
see the
the
picture
special
like this,
brain
that
function
on the
in
surface of
humans
the earth we
which
have
has
the vegetative
really started making
strata,
wonders in
we have got
understanding
the
things
animals,
around
and
us,
we as a human
we
beings, we
not
are
only
also there.
become the the reproductive machinery, but we are also have started
Now,
contributing
if you peep into
into
the past,
development
we
of
can
the nature
really
around us.
understand that
So
how
here, if you see this particular plant at the bottom,
the
a
humans
complete
have
plant,
really
few
made
years
use of
back,
the
even many of us of my
flora on
age,
Earth
more than 50 years of age, would understand that whenever we used to go to the village,
and the
and
kind of
if you look at the tree,
facilities we
our
in the form
understanding
of
was
a
it
look
is the tree which bears a lot of good fruits. It tastes good and it gives
into
wonderful shadow
clothing
when it is
in the various
scorching sun
forms like
outside.
we as a
So that
creature.
was the understanding. आम का पेड़ यानी हमें मालूम था कि भाई ये आम के लिए बहुत famous है. But then with the advent of
The
science and
shelter
by
that
the
he
utilization
creatures,
and
the
evolution of
animals
the
we can say,
brain,
which
its
we go and
understanding, we
choose.
started classifying them into different parts. So this is the
And
kind of
then
primary
available
understanding
biodiversity.
we
The
started
smartness
with our
available in the human
nature.
soul.
We started understanding that, and
If
then
you look
came
into
the body
and
itself,
then
because
more focused
understanding the plant in the form of
understanding this particular capacity in the form of
as
our
a
brain.
food,
Into this,
and
so
then
we can see
utilizing
the
it
picture
for
of
the
this
purpose
on the
of
surface of the
management
we
of
have
the health of the
really
individual
started
was
making
another
strata.
aspect.
We have got the
when
animals around us,
we
we
were
as a
also
human
the
beings,
part
we
of
are
that
also become
particular
the
biodiversity which was given on the
reproductive
earth,
machinery, but
it
we
was
are
also the
also
nature's wonderful
have
creation.
started
And therefore,
if you peep
we
into
need
the
to
past, we can
consider
really
this
understand
body.
that
So
if you see
say where the forces which needs to be properly balanced in our body, and if these five taken together are well balanced in your body, then the three doshas, the vata, pitta, and kapha, are being properly taken care of. It means that whenever it is called as a three dosha, they are not called as the three guna. They are called as a three dosha. Dosha is something when they vitiate or whenever they go out of the control, then it would lead to the manifestation of some kind of disease in your body. So balancing this panchamahabhuta and thereby vata, pitta, and kapha has been the doctrine of principles in understanding and diagnosing the disease in human beings for thousands of years in India. So, amongst the recognized traditional systems of medicines in India and globally, we could see that those systems of medicines of health management, which were having basic doctrinal principles in existence, some documentation in support of it, like Ayurveda has the things in texts, then similarly, we have so many things, and which taken together comprises to become Ayush. Now, these particular traditional systems of medicine have got place in WHO because of their earlier data documentation and the kind of a doctrine of principles in existence, and therefore they have been accepted as alternative systems of medicines and traditional systems of medicines in WHO as well. But let us see this particular slide, that what we had in our legacy. So we had on the other one side some Sahitas written, and some books were there from the Rishis, translated into Hindi, Marathi, Tamil, Telugu, and many other languages. But there is another side of the picture wherein the health management system still more depends upon the experiential basis, and this has come from the generations to next generation. So this is called as a paramparagat vaidya, and there is a process called as a lok swastha parampara. So the Vaidus, or we can say the traditional healer from a particular region, will practice in his region by using the local flora and fauna, and he will start getting the cure to the many diseases for the patients who reaches to them. But then in these two sides, one is organized side where is the documentation talks about the proper plants, their identification, their time of collection, their utilization in the form of a formulation, and the kind of formulations. On the other side, these traditional healing practices are still in vogue, and they are not documented, they are not authenticated, they are not scientifically proven and validated. And these two sides I wish to show it to you before we move further. In modern times, the whole perspective, there was a tree I was telling you about, like a mango tree, and our perception to look at the mango tree was for its fruits and the shade that it gives wonderfully in during the summer days. But with the advent of science, as we started understanding the parts of the plants, then the kind of primary metabolites. Those have been, generated in the plant, and then we understood that there are certain secondary metabolites, or the metabolites which are of some medicinal significance or pharmacological significance are present in the plants. Now, our vision from the scientific perspective started changing. I looked at the now the mango plant in terms of the percentage of mangiferin present in it. So instead of a holistic approach, I started looking into the plant from a reductionist approach. So this is the kind of the teaching and the progress that we started making in last few decades, and therefore we now look at every plant as the source of some or other medicines, secondary metabolites, which otherwise are not made for us by these plants. So why they are synthesized in the plants? There are many good things, and besides them was their own self-protection. So secondary metabolites like alkaloids, glycosides, flavonoids, they are not vital, and therefore they are not present in all the plants in equal amount, or not all the secondary metabolites are present in all the plants, but they are synthesized in that particular plant for the survival of that particular plant. And therefore we, being the smart human beings, we started taking that particular strength of the plant for taking coming out with the cure for many diseases. So this is my picture when I was really mesmerized. While being teacher, I always used to teach my students about the source of
secondary metabolites like alkaloids, glycosides, flavonoids, they are not vital, and therefore they are not present in all the plants in equal amount, or not all the secondary metabolites are present in all the plants.
Morphine and
They
opium is isolated from
are synthesized in that
when I was in
for
Lucknow
the
at,
survival of that particular plant, and therefore we, being the smart human beings, we started
I was
taking
really
that particular
extremely happy
strength
to
of the
touch
plant
upon all
for
this. Now, this
taking
practical aspect of
coming out with the
is
cure for
usually
many
missing in our
diseases.
life.
So this
And
is my
where
picture
the
when
faculty development
I was
program
really
contributes
mesmerized. While
suggest us
being
to go to
teacher,
the
I
fields,
always used
to
to
go and
teach
touch
my
all
students
the
about
plants, to
the
understand
source of
and
secondary
imbibe
metabolites
that particular
like
culture of,
alkaloids,
engaging in
glycosides,
very
flavonoids.
practical
They are not
aspects more
vital,
than the unripe
and therefore they are not
capsules.
present
So
in
it
all
was
the
very easy for me
plants
thereafter
in
to
equal amount,
understand
or
my students about how
not all the
opium is
secondary
extracted,
metabolites are
because I
present
could
in all the plants.
morphine and
Experience
opium
all
is
that
isolated from
are synthesized in that. When I was in
a slide which
no,
shows
the
about
survival
my
of that
pharmacology
particular
laboratory,
and therefore aromatic plant center.
herein we can see that at the center,
The
usually our
smart,
pharmacology sits in
human
the
beings,
center,
we
and then on the
started.
right
I
hand
was
side, the
taking
obvious
that
outcomes
particular
like phytochemical,
strength of the plant
pharmacological,
for this
and
coming out with the cure for many
but
diseases. So this is
I
my
couldn't
picture
stop
when
here because of my
I was
industrial
really
exposure.
mesmerized.
I often entered into the interdisciplinary approach of academia industry, involved many pharmaceutical industry into our work, and we help them in standardization and give quality.
Malpuram municipality.
Hi, somebody is unmuted. So next thing was to expand our horizon, and then we started helping the traditional healers all across India. And I will be now going to Goa for World Ayurveda Congress, where we are holding complete 4 days program for the traditional healers, and then their connectivity with the modern scientists. We are working with farmers, we are working with Go Vigyan, we are working with the forest department, many industries which are now entering into the nutraceutical business, and then we are expanding our outreach by our understanding with the help of the science. So as all of us understand that the plant itself is, the smallest division of it is the active plant cell, and each cell is a totipotent, and therefore it mimics a complete plant. So with the advent of science, we started, growing it in the tissue culture laboratory. And then during my visit to University of Mississippi, I could understand that what is going to be the future of the naturals. So naturals is going to be, highly taken up in future. And here I could, understand what is the approach of the laboratory, which is of the highest standard in the world. So all of them, they have started, looking into the plants right from its basic authenticity. So either they are growing it in the tissue culture lab, or in the open garden, the most important aspect of taking anything for research is the authenticity. So first of all, they have taken this particular care that whatever studies that you are going to perform, you should maintain a proper specimen, and the whole process was properly understood at the University of Mississippi. We started implementing that in our Nagpur University, and many samples now are being stored. Now these are some of the machineries which you need for translating your research into the industrial scale by having the pilot plants at your place, or herein we use the small medium entrepreneurs for with their facilities to translate our research into something which is more practical and more commercial. So at Mississippi, in this side, you can see that from 2001 to 2016, there were so many worldwide approved drugs which were new chemical entities and which came all the way from naturals only. So I wish to tell you all that it would be easier for all of you to work in the domain of research and identify and extract the newer molecules from the natural lab, means from the plants, rather than working in these synthetic laboratories. More so ever with the rider of safety, which is also going to be connected with the molecules coming from nature. So quickly, I would take you to the kind of products which are available in the market, and this is the list of it. And every day, people are trying the new formulations, which all of you must be aware of, and so many products are coming up, which are really attracting the attention of the end user, the consumer, the customer, towards the natural products. So while we as academicians need to establish the quality of the medicine, herbal medicine, we have to be dependent upon the modern aids, and we need to understand the concepts behind the standardization. So these are some of the devices or the instruments which are available with us, and which many of you might be using, but many of you are not using that for that particular standardization process. So herein we have the high performance thin layer chromatography, high performance liquid chromatography, atomic absorption spectrophotometer, or GLC, and each one of them have a significant contribution in understanding the qualities, the phytochemical qualities of the one single plant or its extract or the formulation thereafter. So all this taken together, we as a pharmacy fraternity, we need to really start using these kind of modern aids for substantiating the kind of claims or the research which is related with the herbals. So quickly telling you about the HPTLC. This is very commonly being done in our laboratory, wherein one single extract of one single extract of the plant is dropped at the baseline, and then with the proper understanding of the solvent systems, which can really decipher the number of ingredients which are present inside the particular extract, can be seen on this slide. And it's very easy to actually do the experiment on this, because it uses the we can say destructive modes of observations. Like visually you can see it under the ultraviolet light, and then by destructive means also you can spray it with many detecting reagents, and you can detect and identify a particular constituent present in the extract. This really is a wonderful innovation, we can say, wherein our focus doesn't remain restricted to one single phytoconstituent, but to the spectrum of phytoconstituents which are responsible for delivery of the activity from that particular extract. So we have to appreciate this particular technology and use it while we understand the intricacies or the holistic kind of approach in understanding the phytochemical qualities imbibed into any plant extract. Next, we can move to further critical separation, and then again understanding the phytoconstituents, not only understanding, comparing. Meters were more in organic. O means organic Trigonella phenumbricum, and all these, protein fats, everything was little higher in organic ones. Particularly vitamin C and the energy level was more in organic ones. However, I want to, bring your attention to these particular points, total ash and acid insoluble ash, which was quite higher in the non-organically cultivated Trigonella penumbra. Now, all these factors, how they contribute into the quality of medicinal plants, that we will see. There was a study called as anti-nutritional factor and physicochemical analysis, and this anti-nutritional factors which contributed much are the oxalates and phytates. You could see almost 5 times higher levels of the oxalates and phytates were present, and therefore we started assuming that it's not only the medicinal property but the nutritional and anti-nutritional factors also should be considered while we test the medicinal plants here in the chemically derived medicinal plants. And this study was done in IIT Powai, and this was really an eye-opener for all of us. So heavy metals were higher,
And therefore, we started assuming that it's not only the medicinal property, but the nutritional and anti-nutritional factors also should be considered while we test the nutritional and anti-nutritional factors.
secondary metabolites compared to the
Also should be considered while we test the
we will
nutritional and
go into the
anti-nutritional
quantitative
factors metabolites compared to the
so now you could see the total polyphenols, flavonoids, flavonoids, tannins, all those which we consider as the source of secondary metabolites, and these are the metabolites which are responsible for ultimately division of pharmacological activities. They were all in good quantity in the organically cultivated Trigonella penumbra as compared to the non-organic ones. And this is where the first line of observation which gives us confidence about why it is coming out. Here we can see that in Trigonella phenangricum, we took diazonin as one of the standards, and raised the standard when we estimated
confidence
we found
about
that
why
the organically cultivated Trigonella phenangricum was having better quantity of diazonin as compared to the non-organic ones. So this was estimated by HPTLC as well as HPLC, and we could see them in the form of the peaks. HPTLC was again further taken deep into the quantification level, and we could see that trying to take you to that particular story wherein not only the phytoconstituents but the spectrum of constituents
that
better
the organically cultivated Trigonella penumbra was having better quantity of diazonin as compared to the non-organic ones.
the organic one
So this was estimated by HPTLC as well as
the diazonin or the principal
HPLC, and
constituent.
we could see them in
And,
the form of
we
the
understood
peaks.
that yes, therefore it is needed to grow our medicinal plants by organic means. However, as we all pharmacists enter into the next step after the phytochemicals
HPLC
is the safety and efficacy of the product. So we had a collaboration with Department of Biotechnology,
HPLC, and we could see them in the
at that
form
time
of
we had a very
peaks.
wonderful
Yes,
module
therefore it is
of
needed
testing the toxicity
to
of
grow our
plant
medicinal
extracts
plants
in
by
our
organic means.
which involves
However,
the
as
bioluminescent
we all
bacteria.
pharmacists
Now,
enter
these
into the
bioluminescent
next step
bacteria are
after the HPLC is
amazing
the
because when they are
safety and
alive,
efficacy
you
of
could
the
see
product.
the topmost
So we
phase light.
had a
When they are
collaboration
alive,
with
they
HPLC,
emit,
and we could see them in the form
once
of
they are
peaks.
thrown
Yes,
with a
therefore it is
particular
needed to grow our medicinal
across
plants
their
by
tissue.
organic means.
And
However,
whenever
the
you
bio
use
as
something
we all
which is
pharmacists
detrimental
enter into
or
the
which is
next
toxic
stage
to
after
their
the
life, you
HPLC,
would
pharmacists
and the
enter into
drop
the
in
next stage
their
after
luminescence was
the
clearly
HPLC.
observed. So these are all indicators as to how we need to be careful about the source of raw material. Now, this source of raw material, unless this is
So these are all
of
indicators as
confidence
to
and
how we need to be
unless it
careful
is of
about
organic nature,
the
probably
source
the
of
medicines will not be of much use because they will never deliver the desired efficacy of the pharmacological activity. It was then tested and found,
So these are all
particularly in the anti-diabetic
indicators as to
models
how we need to be careful about
diabetic
organic
studies
nature.
also,
The
the lowest
source of
concentration
will not
you
be
could
of
see that
much use because they will never
of organic one was delivering more anti-diabetic activity than
tested and
double
found
the dose of non-organic antibiotic of the same plant, same medicinal plant, but repeated cultivations were different, and therefore, even at the highest dose of 200 or even 300, we could not see the effect, the anti-diabetic effect on the trichinella and
tested and
so,
found
the outcome from this study is that whenever we as a pharmacist are going to work strong with industry, we need to first ensure or ask the industry to ensure that the source of the raw material is organic or not. If it is not organic, definitely, as the study was showing us, there won't be any positive effect on our health. And this will ultimately lose what? The doctor who is prescribing this medicine, which is made from the chemicals and fertilizers, then there won't be any effect, and the doctor will himself, in the idea the doctor himself will lose confidence in the medicine which has been manufactured by the pharma industry. Now, we being the at the the basics of the research, we need to really educate the industry to use the authentic organic source of medicinal plants only. And then are some other avenues with the advent of science where we are talking about the modern approaches. We know that even the single cell can be metabolically derived, and then through the bioreactor engineering we can extract the same secondary metabolite. Either you can take directly from the medicinal plant stem, or you can derive it from the biotechnological process. But what I believe usually is that these kind of techniques are very cost consuming, and therefore they have not become a popular choice for manufacturing the secondary metabolite. We still are dependent upon the medicinal plants, and therefore let us focus upon the organic cultivation or the organic quality of the medicine. When we started understanding this particular phenomena, we started working with the basic people. Now, basic people are those who are still stuck to their roots, and they are practicing their the curing practices, and they are giving the services to the society at their own level, at their own villages, taluka place, and they are using local resources which are very authentic, very organic, and they are available in their close vicinity. So these traditional healers, we started integrating them since 2016 from the Kolkata conference of World Ayurveda Congress, and I am happy, or I will rather say that I am feeling blessed that I was the person instrumental in making this particular event a success and bringing so many traditional healers from all across India to come and share their own experiences on the platform, wherein the doctors from the All India Institute of Sciences, the ICMR scientists, and the scientific fraternity was listening to them very keenly as to how they claim to heal the broken bones, how they come to rescue of the cancer patients, and many other areas of diseases where it is still difficult for the modern sciences to understand its management. And these are the people who are very appropriately and easily doing that particular services to the society. So, and there were around 150 healers in our first conclave, which was supported by Ministry of Ayush. It took place in Kolkata, and then we went to Ahmedabad, and Ahmedabad in 2018 edition we had more than 200 healers participated. Now we are working very closely. We are connected by heart. They are ready to share us with their formulas, and they wish that the scientific studies and the standardization of their products, along with preclinical studies and the consolidation of their claims by scientific means, can be done now hand in hand. Now, what is our responsibility is to see that their rights are protected, their intellectual property is protected. They should get the access benefit sharing, benefits, so that the researchers should help them, substantiate the science behind their claims and their practices, and it should the outcome of the complete study shall become a wonderful treasure of information and treasure of science for the nation and the particular community from which it is coming. This was one of the conference we had in 2015, a wonderful international conference. Dr. Thirukumaran was part of this, and many of you had been there in Nagpur in 2015. It was attended by more than 20 to 22 nation speakers. And the whole goal was set by Society for Ethnopharmacology, wherein we usually say that dissemination of knowledge on natural health products is necessary, but it has to be globalization of local knowledge and localizing the global technologies. So everything that is good outside can be brought together, and that can be utilized for the promotion and appreciation of our own local knowledge. This is the kind of a service that we are doing by sitting in the position of a scientist, as a teacher, as a researcher, and we want to promote this. Now, I am coming towards the closing end of my talk, but this is something, which is going to really
knowledge.
inspire
This
you
is the
all
kind of a service that we are doing by sitting
to the
in the
gathering
position
of the intellectuals,
of a scientist, as
gathering
a
of
teacher,
the
as a
CEOs of
researcher,
many companies,
and we want to promote this.
pharmaceutical industries,
Now,
as we are going to host the Indian Pharmaceutical Congress in Nagpur during 21st and 22nd January in the year 2023. So this was the conference which was due way back in 2020, but because of the pandemic, we got it postponed for next 2 years. And therefore, my friends, I appeal all of you that do not let any of your students miss this particular opportunity. You should not miss this opportunity, as this is the greatest congregation of intellectuals, academicians, and the industry stalwarts who are going to come up and share their experiences. Particularly when during my visit to Sun Pharma, Dilip Singhvi sir, who is the CEO of Sun Pharma, Dr. BPH Reddy of Hetero, and many other stalwart CEOs, they pledged to come down to IPC for sharing their success stories and for inspiring the young students and the teaching fraternity to enter into a very big way into the pharmaceutical businesses. So I, on behalf of the local organization committee, wish to express my sincere appeal and invitation to all of you to please come to Nagpur. Nagpur is ready to host you in a very big way. I'm quickly going to have this particular teaser for you, and then I will move into the last couple of slides. Just a minute. So please look into this teaser.
Prakash, teaser not visible.
It is not visible.
Yeah, please.
Just a minute. Is it there now?
No.
No? Okay.
No.
So only slide is there. This is the slide.
Yeah, slide is there. IPC Nagpur slide is there. That's all.
Okay.
Prakash ji. Prakash.
चलो किस कंपाउंड होंगे चलो
Participant, just wait. Is back again. I think so. His connectivity is lost. Please just wait. Participant, just wait. Is back again. I think so. His connectivity is lost. Please just wait. Prakash? Prakash ji? Prakash? Your mic is muted. Prakash?
Yeah, now it is okay.
Yeah. Now we are. Yes.
Hi. Yeah, just a
Is
minute.
there anything to?
No,
Please.
so I'm just quickly going to show the arrangements that we are going to have, and besides the strong intellectual input, we are making all out efforts to make this particular conference a big success because of the exhibitions. And today only we have a wonderful discussion and meeting with pharma machine manufacturers. Besides them, the medical devices manufacturers, that is going to be the new arena wherein students are going to see the new opportunities in the coming career. So this is all what we are going to offer to you. And almost 50 CEOs of various big companies, they are going to come up, and I invite all those who are on this call today to come down to Nagpur in a big numbers and take maximum advantage of the academic as well as the industrial connect that you can have in this particular time of IPC. So everybody is ecstatic, and I know that because last 3 years we could not have any IPC or any conferences. Everyone is hungry and thirsty to get this input from this type of conferences. Besides them, you can really plan a big tourisms. You can go to the Shirdi Sai Baba and many other places here. And I'm now with this particular appeal to come to Nagpur. I also wish to share that the early bird entries are going to be closed by 30th of November. So I request all of you to register yourself and ask your students to join for the wonderful pharmaceutical feast that is being in the offing. So I will just end my talk with this particular quote, and this says that whenever we are going to work, we have to work by preserving the holiness, aesthetically extending technological advantages, and developing scientific evidences, and creating opportunities for the generation next. While doing so, we have to keep our heart, mind, and intuition altogether receptive and in harmony. And therefore, I'm now coming down to the slide of my gratitude to all of you, and particularly Savita College of Pharmacy and my close friend Dr. Thiru Malakumaran sir, that the opportunity has been given. But whatever experiences that we had was greatly contributed by so many societies, so many associations, many partners, my industry partners, and our alumni and traditional healers of India, that they gave us the opportunity to explore this particular world of naturals. And when we started integrating the modern approaches to establish the ancient concepts and the ancient sciences attached here with the medicines and their practices, we started really getting the true happiness that we as a pharmacognosist, we as a pharmacist are really contributing for the benefit of the generation next, and protecting the intellectual property that we had from thousands of years amongst our fellow traditional healers. So thank you so much for joining us here, and I again appeal all of you to register, and all of you are welcome to Nagpur. Nagpur is gearing up very nicely and very strongly. Welcome you all. Thank you so much.
Vandana madam.
Yes, sir. Thank you.
Please.
Thank you so much for enriching our knowledge through your session, sir.
Thank you so much.
Once again, thank you, sir. Dear participants, if you have any queries, just raise your hands or text in the chat box.
I will
Yes,
just quickly
Deepa
tell you
ma'am,
that.
you raised.
I will quickly tell you the website which you can note down, or you can just Google it and click it. It is www.ipcnagpur.com. It's very easy. And since all of you are from different places, you know that Nagpur is at the heart of India, the city of oranges and the city of tiger. So you have so much of natural resources to visit here, including you can sight the tigers as well, very close to the Nagpur city. So all of you are welcome. Thank you. Any queries? I'm ready to answer.
There are two hands are raised.
Yeah, please.
Subhashini, please unmute your mic and you can ask, please.
Yeah, Subhashini is there.
Roma guys. Can you please
Sir, can I
ask
ask
your
one question,
questions?
sir?
Yeah, Roma ma'am, please.
Yes, sir. So first of all, thank you, Dr. Suhas, for a very
Now
nice
I
presentation.
request
Especially,
Just a minute,
you
please.
told us how organic part is
Sujata ma'am,
much
deliver the
better than the activity grown. Sir, I just want to ask two things. Like we are having simple HPLC in our college. I'm from Kite School of Pharmacy.
Yeah.
So basically, I just want to ask,
Okay.
we have simple HPLC. So you told us like HPDLC can give us proper bands and can help us in differentiation
ma'am.
of the phytochemical constituents present in a particular extract. Can we do the same with the same HPLC, normal HPLC?
Yeah, you can do that. See, in HPLC, what remains significance is the column that you're using, number one. And second is the choice of solvents that you are going to use. For this also, you should first, because HPLC is a very sensitive process. And as I told you, first thing was the selection of proper columns. Second was proper solvent systems. And third is the kind of a detector that you have. So particularly while working with natural products, you should have the PDA detector, photodiode array detector. And only the UV detector will not be sufficient one, because many of our phytoconstituents, they are not in the range of UV lights. So it is most likely that many useful phytoconstituents may go undetected if you do not have the PDA detector.
Okay, so photodiode detector should be used along with it.
Used, yes.
Fine, sir. So another question is, sir, like you said, you did some biotechnology activity.
Yeah.
With your Trigonella this particular
Trigonella fenugreek.
meeting.
Yes.
Yes, sir. So, sir, basically, are there any such labs who are doing this tissue culture samples and all, because it's very difficult for a college
Yeah.
to keep all this aseptic conditions and basically do this anti-cancer activity or any type of this cellular activity.
Right.
Could you, yeah, could you please recommend us some labs so that we
True.
can do such activities?
True. There is always a kind of a question in everyone's mind there, and as well as that kind of a question was there in my mind, that I have only limited number of instruments in my lab, and how can I do big research? So
Very
at
true.
that particular time, the best thing is my own connect. Like all of us are now connected together here, and like my friend Thiru Kumaran is there in Chennai, and what we did was we started going to the conferences first. We started connecting with the right individuals with their own strengths, and we started connecting each other's strength. Like I have some qualities, some instruments, and some outcome from my laboratory, but I use the biotechnology lab of Jamia Hamdard in Dr. Saeed's laboratory. I use Professor Pulok Mukherjee's lab for atomic absorption spectrophotometer or GCMS. I use Dr. Sanjay Yajak's laboratory in Naipur for all high-end analysis like LC, MS, MS, and above that. So the question doesn't remain that whether I have all these equipments in my lab or not. I have wonderful friends, I have wonderful connect, and just on a phone call or a request on email, my sample once goes over there, they will do the analysis. And what else we want? We want the results from the analysis, which we need to collectively interpret and then publish it or make some valuable, patent out of it and something like that.
So
So,
that that means we have to
so
make contacts.
contacts, and that is where we boast upon that this IPC, we are going to have almost around 10,000 to 12,000 delegates in Nagpur. And why are people coming to us? Because we have been connected with them for so many years together. Almost 20 years we have been working with each other. So all big experts, almost 25, foreign, our, collaborators from University of Pennsylvania, Dr. Henry Daniel coming, who is, doing disruptive work on developing the oral insulin in the plants. So a disruption is in this particular technologies, you have to eat the leaves of the plants, which is going to, release and biosynthesize insulin in your body itself. See, that is a kind of a disruption which we all must understand. And for them, for that particular matter, our contacts with Dr. Henry Daniel has worked. He has agreed to come down to Nagpur IPC. Dr. Yashwant Pathak, who is a topmost personality in nanotechnology. He is coming from University of Florida. Now, how these people are coming is because of the sharing of, concerns with each other. And that is where you have to come, because you have to pick the people of your choice, make them into your network, and start making big, even from a smallest college or the remotest college. Anyone can do big research just by connecting with the right people. That is what my answer is.
Right,
Ma'am,
sir.
Roma
I got
ma'am,
yeah.
Roma
Right.
ma'am, that's
Hanji.
what
Hanji.
the same way I got contact with the Nagpur, Jamia Ahmedabad,
Yes.
and then Ahmedabad, and then Jadavpur University. So because of see, I used to go these places always. I used to visit. I don't bother out my money. I
Yeah.
used to go and I made contact with them. That's what I invited him for this lecture, ma'am. So please
Right.
make it.
Very resource person, so the very useful resource person you have invited, I must say.
Yeah, that's what. That's why I'm also roaming around. Not only India, China. I visited along with these people. They helped me a lot. So like that only you can make it good contacts.
So many of the times, sir, we as a faculty member, we usually complain that we submit proposals to DST, DBT,
Yeah,
UGC,
correct.
and we
Yes,
don't
yes.
get the projects, right? What are these projects are for? We usually ask one instrument, and we wish to finish one small project, isn't it? But then,
Yes.
if I need, say, NMR 10 times in a year, and NMR is costing almost 2 crores rupees, and if I'm asking for a project to any big concern, they will say, how much is the utilization of that? And for that, writing that project, how much energy I am putting into. Instead, I am taking help of industries. They are supporting my students because they are working on the research questions coming from the industry. So industry is supporting my student. My student is carrying out work in the world-class laboratories, in the world-class, on the instruments, and the best outcomes in the form of the presentations, publications, patents. Yesterday only we filed 3 patents on a wonderful research that was there from the fish oil. See, imagine Nagpur is not having sea around, but we are working on fish oil, and we have extracted wonderful molecules which are behaving in preclinical as well as the cellular studies wonderfully well. Now we are taking them to the industries in a very big way. So everything is possible sitting at our place. No problem at all.
Got it, sir. Thanks, sir. Thank you so much, sir. Thank you so much for informing.
And all of you, we wish to see you in Nagpur.
Sure. We'll be
Sure, sir. Thank you.
Subhashini madam. Subhashini madam. Okay. Vandana madam.
Yes, sir.
Please go ahead.
Shall I continue, sir? Now I request Mrs. Sujata ma'am to deliver the vote of thanks.
Thank you, Vandana madam. And I thank our Chancellor sir and Director sir and Registrar sir for providing this facility to conduct this 5 days interns faculty develop program. I thank our advisor, Principal sir and Vice Principal sir for continuous support in organizing this FDP. And I would like to thank organizing committee, Dr. Thirumalai Kumaran sir and Vandana madam for conducting this wonderful 5 days intense
Advisor, Principal
FDP
sir
program.
and Vice Principal
thank
sir
all the participants once again for
support in
participating
organizing
in this FDP. And we successfully completed our first day national FDP program. And another remaining 4 days, we all expecting our participants to join in this program. Thank you, sir. Thank you all, sir.
Thank you so much, ma'am.
Thank you, ma'am.
Sucharita madam.
Yes, sir.
You missed Prakash sir also. For same time.
Okay. Okay, sir. Thank you. Sorry, sir.
No problem. Okay. Thank you.
Yeah, thank you, everyone. Thank you so much. Thank you, sir.
Thank you. Thank you, sir.
Participants, Madam Vandana madam, she sent
Yes, feedback
link
form.
Please fill it up and send it. Those who are filling the feedback form, they will be getting the certificates. Please.
Yes.
Okay.
By this, we can end this
Thank you,
session.
Prakash. Thank you.
Yeah, thank you so much.
Thank you.
Bye bye. God bless you all. God bless you all. Bye bye. Thank you.
Thank you. Man.
Your email ID, if we can get.
Yeah, I'll text it here in the chat box.
Yeah, please. Ma'am, I'll send it to you. Don't worry. Roma, ma'am.
Thank
I sent it. I sent it.
It is there.
got it.
Right. Thank you so much. See you all. Bye. Take care.
Thank you,
Thank you, Prakash.
Thank you
Prakash.
so much, sir.
Vandana madam, thank you.
Yes, sir.
Sucharita,
Okay, so thank you, sir.
Thanks a lot. Sucharita
Thank you,
madam, our principal
Thank
sir,
you. Thank you,
our
sir.
faculty members, those who participated and not participated. Thank you. We can leave, ma.
Yes, sir. Day 2 session will be continued on Monday, 6 p.m., sir.
Okay, thank you. Thank you.
Thank you all.